Cloning is a method of producing genetically identical organisms. A clone is a collection of genetically identical copies. Asexual reproduction in both plants and animals result in clones, or offspring, that are genetically identical to their parents' organisms. Humans have been artificially cloning plants for centuries, using many methods. It is only recently that it's been possible to clone large animals. Cloning raises ethical, social and moral questions for many people, and the issues are still being debated. Human reproductive cloning is illegal in many countries, including Australia. Therapeutic cloning involves the use of stem cells. These are cells that have the ability to develop into specialist cells. Stem cells are found in both embryo and adult, but in adult they are restricted to certain areas such as the bone marrow. Therapeutic cloning uses genetically engineered techniques to produce lines of cultured stem cells. Most therapeutic cloning is for human use in medical therapy for treatments for injury and disease. These include Parkinson's, Alzheimer's, spinal cord injuries, heart disease, diabetes, burns and arthritis. In the future, there is the potential to produce complete organs for transplant using this technology. Stem cell research is still controversial, and in Australia, there are strict controls over the use of both human embryos and adult stem cells for this purpose.
PCR was developed in the 1980s. It stands for polymerase chain reaction. This is a process which selectively copies a specific DNA code from any source hundreds of millions of times in an hour. DNA polymerase is an enzyme which catalyzes the creation of a template DNA strand. It's very handy for copying DNA, which is not available in large fragments. Let's go through the process step by step. First, the DNA is heated to 95 degrees. This breaks the weak hydrogen bonds that hold the DNA helix together. This is known as denaturing the DNA and creates a single strand. Next, the DNA is cooled to 45 to 72 degrees. We use a primer in this step, which is a small section of single-stranded DNA, usually 20 nucleotides long. The primer binds to a specific region on the DNA that is going to be copied. It specifies the DNA sequence that is going to be copied and initiates the replication of it from that point. The reaction is then heated to 72 degrees, which is the optimal temperature for DNA polymerase. This is the step where our magical enzyme really starts to work. It extends the primers, adding nucleotides onto it in a sequence based on the template DNA. That's it. Now the whole process repeats itself about 20 to 35 times. The process is exponential because the DNA made in previous cycles can serve as a template for the next reaction, and so on. Now let's take a look at what PCR can do. In 1999, DNA was taken from a female Tasmanian tiger pup preserved in ethanol for over a century. In 2002, these genes were successfully replicated using PCR. These short fragments were undamaged and possibly functional if introduced into a living animal cell. Unfortunately, the project was abandoned in 2005 due to lack of facilities and skill to further the cloning process.
There have been a huge amount of organisms cloned or attempted to be cloned in Australia. However, only a handful have been successful. Some of these include the Tasmanian tiger and the Wollemi pine. Tadpoles were attempted in 1958 using nuclear transfer from a different cell. This was unsuccessful as none of the tadpoles transitioned into frogs. It did, however, show promising signs for the future.
Dolly the sheep was cloned in Scotland in 1997 using a technique known as nuclear transfer technology. In 2000, Australia's first cloned animal was born: Susie, a Holstein cow, and Matilda, a merino sheep.
The Wollemi pine is a plant thought to be extinct and only known previously from fossil records. It has been saved through cloning after a tree was found in the Blue Mountains in 1994. All of the plants currently for sale around the world are clones from the original trees found in the wild.
Created using Powtoon.